Okay, let's go ahead and get started. Good afternoon. My name is Olga Vitek. So I'm a statistician by training, interested in all things design and analysis of quantitative mass spectrometry-based experiments. And today we have 2.5 hours, I think, pretty substantial time to talk about two things. And so the goal of this 2.5 hours is to do two things. One is talk about the design of experiments, and the other talk about the analysis. And the first part, I will talk for quite a while about the experimental design, because I find that this is the most important part of the study. Because, you know, if you analyze the data incorrectly, well, in the worst case, you can go back and reanalyze, right? But if your experiment is designed incorrectly, that's pretty much the end of the story. And I find it's extremely important to spend time on that. And what I will also do, I will talk about some basic statistical summaries, and probably some of you will be very familiar with those, but it is very important that we're all on the same page. So if you have seen these things, just bear with me, and then we'll talk about analysis methods, which will be more advanced, kind of more appropriate for mass spectrometry specifically. Okay. So the first thing, why do we need to have a whole afternoon on talking about statistics? Well, and the point is that in the type of experiments we talk about here, the variation and uncertainty, these are the hallmarks of the studies. And this can come from multiple sources. So we can have technical variation, for example, how the samples are stored, processed, and so on. Instrumental variation, the type of mass spectrometer, you know, the chromatography, ion suppression, and so on, definitely impacts the intensities of the peaks. Signal processing, and I will talk about this a little bit later, also introduces some amount of uncertainty in terms of different tools, have different strategies for locating, quantifying, identifying peaks, and that also contributes to discrepancies in the results of the analysis. But all of these things we can potentially overcome if we design experiments correctly and if we're careful with the analysis. However, the most important source of variation is actually not all of this, but it's really the biological variation. So the purpose of our experiments is to study living organisms, right? So study living systems. And the fact is that there is a natural variability in protein abundance from one person to another, from one time point to another. And when we are interested, for example, in relative quantification, so we're interested in studies of changes of protein abundance between conditions, we want to know if the change is larger than the random biological variation is expected. And so even if we have perfected our designs and measurement technologies, we still have to essentially work with statistics, because we need to understand how much biological variation is in our studies. And so you can think of statistics as a body of methods for making wise decisions in the face of such uncertainty. And really the reason why we need to use statistical methods is to make sure that our research is, on one hand, reproducible, but on the other hand is also accurate, which is not the same thing, right? And so statisticians contribute to this in three ways. So the first way is the experimental design. How can we allocate our experimental resources in a way that avoid systematic mistakes, but also in a way that makes effective use of available resources? So how can we essentially get more information with limited constraints on resources? And then data analysis. So once we collect the data, we actually want to analyze the data in a way which gives us objective conclusions. And the last thing is the statistical tools, which would allow us to fully automate this process. And the reason why automation is important is not because we have large data sets, but it's because if the analysis is automated, it means that somebody else can repeat it and reproduce. And it also means that the decisions we make are not dependent on our mood of the day, you know, or something like that, but we know the specific rules by which the decisions are made. And this is extremely important for reproducible research. And so statisticians contribute in these three ways. So first, before I even talk about the design and things like that, I would like to spend some time talking about how to translate a biological question or a clinical question into a statistical question. And this is extremely important, because I noticed that sometimes people say, you know, I have this really interesting samples. I just want to run them on the mass spectrometer and see what happens. Well, I can tell you up front that not much will happen, right? If you don't have a specific question, if we don't know why we do the experiment a certain way, it is very unlikely that we will find something meaningful. But then to design the experiment correctly, we need to translate this biological question into a statistical question, because different statistical questions will require different methods, and different methods will require different designs. And so we really need to think about all of these things upfront before we even start working with the biological samples. So let me spend some time on that so we can classify statistical goals broadly in 3 groups. And each of the groups has something to do with the class. So class means some collection of samples or collection of proteins which share some similarities between them. So the first goal is class discovery. And you have all seen figures like this, for example, where we have the rows will be proteins, the columns will be biological samples, and the colors are the abundances of the protein in the sample. And we would be interested in finding patterns, right? And so we can say, okay, what about proteins which behave similarly across samples? Well, this may be meaningful because maybe these groups of proteins are involved in the same biological process, right? Or maybe we want to look for samples which are similar in the space of proteins, which is also meaningful because maybe this is a subtype of a disease which we discover that way and so on. And of course, there are also some clustering of profiles or principal component analysis here. Each axis is a linear combination of proteins. Each point is a sample, so samples which go together are very similar. So the important property of this type of methods is that they do not give you upfront the definition of a class. It just says, okay, go and find something similar. And there is no definition of useful similarity versus nuisance or not useful similarity. And so the methods will go and find something, but how likely we're to find something useful, it's another question. Because usually things are similar a lot for technological reasons, for example, batch effects or some kind of processing issues and so on. And those things will appear kind of similar on these plots and on these plots. So these methods are called unsupervised because you don't have a class label that is associated with them. And these methods are super nice for data visualization, really nice for quality control, really nice just to summarize kind of your data initially. They are not good for doing really any particular biology. So very rarely you would find methods which are completely unsupervised useful in practice. They are certainly not good for clinical studies, for example, for finding biomarkers of disease. So that's very important. Now the second statistical goal is class comparison. So now this is a supervised method in that we have 2 classes of samples, healthy and disease, right? Or disease 1, disease 2, and so on. So now the method actually is aware of which sample comes from which class, and we're interested in which proteins are upregulated or downregulated between conditions on average over all the samples. And so here's one example. So let's say we have one protein here. These are healthy subjects. These are disease subjects. And we want to know if on average over replicates, the protein went up or down. And the methods you would use would be t-test, analysis of variance. The readouts would be p-values or posterior probabilities or something like that. And this is very useful. It is very useful in basic biology. It is useful for discovery of, potentially useful targets for subsequent targeted follow-up. A common mistake that people make is that when they find a protein which is regulated and let's say it has a small p-value, then people say, I found a biomarker of disease, which is wrong. Because again, this type of methods are not designed to discover biomarkers of disease. And the reason for that is that, again, we need to think about how the clinical, in this case, question translates into a statistical question. So the clinical question is not about, does the protein change on averaging? If your problem is discovery of biomarkers, the context of that is the biomarker is supposed to be actionable for a clinician, right, for a particular patient. So the clinician is not seeing an average patient. The clinician is seeing a particular specific patient and needs to make a decision for this patient. But here the statistical goal is to compare averages. So this is not necessarily actionable. Therefore, this method is not necessarily good for discovery of biomarkers. And this is something which really is important to remember. So here's just some examples. So here you can see, let's say, healthy and disease, and we have subjects very tightly clustered around the means. And so if the doctor sees a patient with this protein abundance, it's likely coming from the disease group. So this is a case where it's regulated and predictive at the same time. But here's another situation where you have exactly the same difference between the groups on a log scale. but you have so much variability in the population that if a person comes with this protein abundance, it can be any of the two groups. So it's not necessarily a well-predictive protein. So here it will be differentially abundant, but not predictive. So this will not be a good biomarker, even though it has a small p-value. And the opposite is true as well. So here is the case of two proteins. Let's say blue dots are healthy people and red dots are disease. So here we see that protein 1, for example, has low abundances for healthy, high abundances for disease. Clearly it's regulated, right? The same thing for protein 2. So we'll have small p-values, and we can clearly predict the status of the disease for each subject. But here's the opposite situation. So you see that the healthy subjects, they span the full range of abundance of protein 1, and the same thing for the disease subject. So if you compare mean abundances between these two groups, you will not have a change in the mean. The same thing for protein 2. But as a bivariate panel of two proteins, they are perfectly predictive. So essentially, class comparison is only comparing averages. And so if the biological question is asking for averages, then it's a really good approach. But if the biological question asks for the status of individual patients, then it's not a good approach. So that's something to keep in mind. And so if we really want to do biomarker discovery, this is more of a statistical goal 3, which is class prediction. And so now we focus on predicting the status of individual subjects. And so what it would do, well, for example, here we have two proteins. So essentially we will be developing rules or decision boundaries which will say which subject is healthy and which subject is a disease. So here, for example, we ignore protein 1. We use a cutoff of protein 2. And we say high abundance is healthy, low abundance is disease, right? And it's kind of okay, but we also have a lot of misclassified cases. Now we have a more complicated rule which uses both. Now it's doing a little bit better. Now we have a very, very complicated rule which perfectly kind of separates red and blue. But this is probably accommodating just too well, some artifacts that we have in data acquisition or data collection. So probably this particular decision boundary will not be reproduced if we were to repeat this again. So this is called overfitting. And so usually these methods proceed by looking for this type of boundaries, looking for one which is not too simple, not too complex, kind of in between. And the readouts will be very different types of error rates. So you'll have sensitivity, specificity, positive predictive value, precision, recall, depending on what you do, right? So this will be, and it would require very different methods. It would require logistic regression, random forest, support vector machine, deep learning, whatever you would like. So this will be a very different class of methods. And therefore, they would require different strategies for experimental design. So I think, and I see quite a few of you are familiar with this concept. So again, I hope I'm not boring you too much, but importantly, we need to translate the biological question into this type of framework to see how many replicates, for example, we need, or how to allocate resources appropriately. And I will give you some examples of that. So that's really the idea. Any comments on that so far? Okay. All right. So now let's talk about experimental design. And again, these are basic concepts, but I am surprised how often I see that people forget that or think that for some reason things don't apply. So the fundamental principles of experimental design, of statistical experimental designs, apply very much to quantitative mass spectrometry and proteomics. So here is what happens in my mind when I think about an experiment. And by the way, for a large part, let's focus on this second goal, where we say we want to compare averages, right? So this is kind of the intermediate goal, which is frequently of interest for us, right? So we compare the regulation on average of replicates. So if we have, let's say, an initial clinical study where we just want to see, okay, what are the good targets for follow-up? So this is what happens in my mind when a collaborator comes to me. So, and let's just say health and disease because it's easy to talk about, but you can translate this to wild type and knockout or anything else you want. So we have two populations. These are healthy individuals, and this is the second population, which is the same from the point of view of, age, gender, lifestyle, other diseases, anything that can affect protein abundance, except for the fact that this population has the disease and this population is healthy. So this is already a fundamental concept that we have two populations which are infinite of infinite size for all practical purposes, and they have the same properties except for the status of the disease. So because the populations are infinite, and we want to know which proteins are different on average between the two populations. So since the populations are of infinite size, we cannot quantify protein abundance of each subject in the population. What do? We select some subjects into our study. And because we select some subjects randomly, and because there's a natural variation of protein abundance between the subjects, if we were to do it again, if we repeat our experiment, we will have some other subjects. So this is the first layer of variability, that from one replication of the study to another, we will not have the same subjects. Therefore, they will not have the same abundance. But this is not the whole story, because even though we included some subjects in the study, we still don't know what their abundances are, right? So what we need to do now is actually quantify the abundance of the proteins, which, with the technology, which for the moment itself is subject to, noise and uncertainty and variation. So this is the second layer of variation, which is technical variation. And this is the only thing that we actually observe, right? So this is biological replicates, these are technical replicates. And so the goal of statistical inference, and this is really how statistical inference proceeds, is to take what we see and use it to make conclusions about what we really want to know. So our peak intensities here in the selected subjects are only useful to us as much as they're informative about the actual broader underlying populations. And so now, if we take what we see and we want to make the conclusions about what we don't know, there are at least two ways where things can go wrong. And these two ways are bias and inefficiency. So bias means that we are systematically wrong, right? So our conclusions we make from what we see deviate systematically from the truth. And bias is the worst kind of problem, because bias is reproducible. So if we design the experiment incorrectly in a way that we have bias, we will see the same bias again. And we will say, great, we reproduced our results. Well, we're reproducibly wrong. So this is not very helpful, right? So what we really want is to make sure that overall our results are informative of the truth. And the second problem is inefficiency. And inefficiency means that what we see is so noisy or has so much variation and uncertainty that for all practical purposes, we cannot say anything useful about the populations. And so the goal of experimental design is also to minimize inefficiency so that our data are as informative of the populations as possible. So this is really the idea. Now, I won't do much mathematics, but I just want to show you a little bit how statisticians think about that, so that if in the future, if you see this, you kind of know what's going on. So the convention in statistics is that we use Greek letters for things which are not observable, and we use Latin letters for things which are observable. So we have these constants in the populations where they are the mean abundances of the protein in the entire population. So this is a constant which we don't know what it is. We will never ever know what it is, but it's out there. And so we assume it has a number, right? And the same thing for the disease. Now for the subjects, they have their own constants, which are the true protein abundances in those samples which we selected. Again, we don't know what they are. What we do see is the intensities of peaks in mass spectra, right? Maybe MS1 peaks, maybe many fragments or transitions, many peptides. And so the goal for us is to take what we observe, summarize this in some useful ways, and describe the statistical properties of the summaries. And this allows us to make some statements about the underlying populations. And so bias in this case means that our differences are systematically away from the true differences. And the inefficiency means that we have large variability associated with them. So, by the way, let me just kind of comment here. So Andy talked in the morning, he talked a lot about ratios, right? And I talk about differences. So how does it fit together? Actually, we're talking about the same thing, because everything that I will be doing here is on the log scale. So the very first thing we will do is to take the log of peak intensities, and the log of the ratio is the difference of the log intensities. So whenever I say difference, think of this log ratio between conditions. But on the log scale, things have much better properties for statistical purposes, and that's why we do that. Okay, and so bias and inefficiency can be prevented by three fundamental principles of experimental design. These principles I did not invent. They exist for like over, I don't know, like early 19th century or even before, right? And they have been applied in many areas of, it started in agriculture and so on. But what I noticed is that every time there's a new technology out there, something really high throughput can quantify a lot of things, and very exciting, then somehow people forget that these things hold just the same, right? And so this is really important for us to remember. So let me review that, because this is extremely important for us. So the first fundamental principle of experimental design is replication. And I hope I do not need to convince you that we need to have replicates, right? So, but let's see why, right? So let's just have some really simple example here. Let's say we have some LCMS experiment where we have one peak per person, right? Let's say one healthy and one disease. So now if I just make this plot, healthy and disease, this is the log intensity of this feature, log abundance. So this is the log fold change. So is it systematic or is it a random chance? Well, exactly, who knows, right? It's absolutely impossible to say, because we don't know how much randomness can be expected from that, right? So now here we actually have biological replicates, which are very tightly clustered around the means. And so now we see that this change is unlikely to be an artifact of random variation. But now we have exactly the same change, and we have a lot of biological variation in each group. Now this is can very well be an artifact of random variation. So exactly the same log fold change may or may not be systematic. So consequence of that, by the way, is fold change cutoffs make no sense whatsoever, because exactly the same log fold change may be an artifact or may not be artifact. So we really need to have replicates to know how much variability is there, so it allows us to carry out the inference. But also, well, it helps us minimize inefficiencies, because if we have more replicates, we are really, really confident in this mean, really, really confident in this mean, and we can claim that this change is systematic. And so because we have two layers of random variation, we have biological replicates and technical replicates, and so we can potentially have both, right? And I'll give you the upshot. Biological replicates are the most important. So if we have one sample and we have 10 mass spectrometry runs on this, we quantify this sample really, really, really well. And we quantify maybe another sample from the disease group really, really well. But it's still one sample. So we have no idea how much person-to-person variability will be there. So if you have a fixed number of runs, you are much better off allocating one run to each new biological replicate. And there are kind of mathematical techniques to demonstrate that, which I'll probably not have time to talk about. Okay. So this is for replication. So the second fundamental principle is randomization. So randomization, this is something which is unfortunately overlooked a lot, and the purpose of randomization is to prevent bias, especially bias from artifacts that we are not aware of. So this is an example. So let's say we have healthy and disease, and we have 4 replicates of healthy samples which were all acquired on the first 2 days of the study. And these are 4 replicates of disease which were acquired on the next 2 days of the study. And so now we see that the difference is unlikely to be an artifact, but the problem is that this experiment introduced a confounding, because now this difference can be due to at least 2 sources. One is the disease, but the other is the time of data collection. And if you want to guess, at least my experience indicates that if time of data collection is an issue, it's always the time that is responsible for the difference and not the biology. So these experimental artifacts, they always are stronger than the actual biological effects. And so essentially, this is an example of an experiment which is confounded with an artifact, and it's a wasted time and money. Now, what we can do instead is to allocate randomly each experiment or each sample to different days of acquisition. So let's say we have something like that, right? And so we randomly allocate different samples to different days. So now we have a lot more variability among healthy and a lot more variability among disease, because now this variation comes from 2 sources, biological variation and day-to-day variation, but at least the days are kind of roughly equally allocated between the 2 groups. So on average, we don't have the confounding anymore. So randomization, especially in long, kind of large-scale studies, allows us to prevent this kind of additional biases which are unrelated to our question of interest. Does it make sense? Okay, and again, because we have two levels of randomness, we have two types of randomization. We need to select randomly biological replicates from the population, and we need to randomly allocate samples to all steps of sample manipulation and data collection and all of that. So just some examples. This is a paper which I really appreciate, kind of old, but still speaking to the same point. So these are statisticians from the MD Anderson Cancer Center. They are working with their clinical colleagues in data analysis. So this was quite old. So this was a CELD experiment. It really doesn't matter. So they received these samples from 5 types of cancer, and they were blinded. So they didn't know which sample came from which type of disease. So they did whatever was the state of the art at that time, normalization and so on, picking. And they did class discovery. So this is the first goal, right? So they tried to cluster samples, and they found more than 5, looks like 6 subsets, right? So 6 subgroups. And they were excited, thinking maybe we found a cancer subtype or something like that. And now they made a second plot where the x-axis is exactly the same as here, but the y-axis is the day when the samples were collected. And you see that these clusters correspond perfectly to the time of data collection, as opposed to the biology. Now, it turned out that the first 2 batches were one type of cancer, and the next batches was another type of cancer. So this is a wasted experiment. So these statisticians, they published several examples of essentially studies which failed like that for reasons of design, so that we can learn from that. Okay, so then there are additional examples. There are many in the literature, and this is probably my favorite kind of, bad example. So this comes from the blog by Jeff Leake, who is a statistician at Johns Hopkins. This was a study of gene expression between Asians and Europeans subjects. And they found that 78% of genes are differentially expressed between these 2 groups. So 3/4 of the genes are differentially expressed, right? Kind of crazy. And so their colleagues came and asked to see if they could repeat the analysis. Maybe there was a typo, or something wrong. So they went through, and they didn't see anything wrong with that. So all the things they did were kind of meaningful. And it really took them to go to the very raw data. So at that time, it was done with Affymetrix arrays. And luckily, the very raw cell files of the arrays were publicly available. And so the Affymetrix arrays, they have a timestamp in the corner, like in the very kind of raw data themselves. And so it took going there to realize that there was 1 year of difference between when they collected samples on Asians and samples on Caucasians. Well, no wonder that many things are different, right? But they're different for wrong reasons, right? And so this has nothing to do with biology. So this, and when I say I'm surprised how often it happens, it happens, to the date. And in my experience, we had some collaborations where people forgot to randomize, or saying, it's too expensive, or it's too complicated or something, and we would insist. And then the experiment would be redone, at least on the technological side, and you see how different the results are between the randomized and non-randomized versions. So these are really important things to remember. Okay, so this is for the for this. And this is like one of the popular quotes from Fisher: "To call in a statistician after the experiment is done may be no more than ask to perform a post-mortem examination. He may be able to say what the experiment died of." Right? So this is kind of what is happening, right? Okay. So this is principle number 2. And I would say that having replicates and randomizing, it's probably something that everybody can do, right? So with care. Although one thing we need to mention is that when I mean randomize, I mean actually use computer tools, right, or software to establish random order of data acquisition in the samples, or randomly select samples from the fridge, as opposed to do that, And the reason is because humans are terrible at randomness. We actually have no sense at all of what random actually is. And so there are many examples kind of on the sociology side where what we would end up doing is not random at all. And so it is really important to use even simple tools like in Excel or in R and have randomization. So I read a study at some point. So they were studying mice for something, I forgot for what. And so they had to take some mice and apply some treatment to them, and then the others will be controls. So they try to actually grab some mice randomly out of the cage and then apply treatment, and the remainder were the controls. Well, turned out that they could grab the mice that were slower. And that affected whatever conclusions. So they're really silly things like that, which can really affect the results. Okay, so the third fundamental principle is the one which would require most of a statistics expertise, and this is where I would really suggest consulting a statistician as opposed to just doing something on your own. And this third principle is blocking. So blocking actually has both purposes, reducing the bias and reducing the inefficiency. It does both, provided that we know a potential source of variation. So let's say we have a potential source of nuisance, such as day of or time or batch of data collection, right? Or maybe some other batch of materials, right? Or something else. And we know it exists. There's nothing we can do about it, but at least we know it, right? And so this is my previous example where I would randomize the samples to different days. And so now if you look at my figure, you see that here at day 4, both healthy and disease are kind of higher, right? And here at day 1, healthy and disease, they're kind of lower. So if we know that time of data collection is an important factor, what we actually can do is to impose restriction on randomization. So blocking is a restriction on randomization, where we say on day 1, I forced to have 1 healthy and 1 disease. Now the healthy I'll select it randomly of all healthy. Okay. And disease, I will select it randomly from all disease, but there will be 1 of each on day 1, right? I will do the same thing on day 2, the same thing on day 3, and so on. And so now what we can see is that we have created a block where we restricted randomization separately within each day. And what we can do, we can consider within each day the difference between healthy and disease. And so here we see that within the same day, the difference always points in the same direction. And so now, instead of comparing the mean of this group to the mean of this group, I can only focus on these differences and ask if the difference is systematically going in one direction or the other. And that allows me to essentially eliminate the day-to-day variation. Like that. And well, here my example is that I have 2 samples in a day, but there are statistical techniques which can have any number. I just can't draw a picture as easily if we have more days. And so this is what is called blocking. It requires 2 things. It requires us to know that we have this potential batch effect and design the experiment accordingly. So forcing randomization within a day as opposed to do things randomly. And second, it requires us to analyze the data with the understanding that there was blocking. And so therefore, it's really important to know how the experiment will be analyzed to decide how to allocate the resources. And because we eliminated day-to-day variation, you can show mathematically that this type of blocking design will require fewer number of replicates than the design which is completely randomized. And so these are really useful techniques for us to know. So, you know, this is how statisticians can help. So people sometimes say, I don't like to work with statisticians because they ask for replicates. Well, we do, right? But at the same time, we can also, we're not just like saying, you know, give me like 5,000, although I will be talking about 5,000 very soon. But at least with some limited resources, there are techniques to reduce the required number of replicates because we understand that the resources are limited for everybody, right? And so because we have, again, 2 layers of variation, biological variation and technical variation, we will also have 2 types of blocking. Blocking on biological characteristics and blocking on technical characteristics. And so, well, here's, first of all, for the technological characteristics, an example, again, from the same paper. Also, they had some cell-death experiment with 2 types of cancer. Again, blinded, they found 2 groups, looked very good, except that when they unblinded, they realized that this was a protocol change, which, of course, will be the main source of discrepancy in the data. Now, if this protocol change is unavoidable, maybe because, we need to do some maintenance in between data collection, then we can plan this in advance and make sure that we have enough healthy and disease before the maintenance and enough healthy and disease after the maintenance, and then combine these results. And so this will be a good use of the experimental resources as opposed to, completely randomized. And so blocking with respect to biological characteristics is referred to as matching. So here's an example. So if we have our populations, which are quite heterogeneous, especially if the number of replicates is fairly small, if we completely randomly select subjects, even if we do everything right in theory, we may still end up with healthy who are, I don't know, older and male, and then disease who are all younger and female or something like that. So blocking would impose a restriction on these potential confounders and say, I want to have one younger female from each group. I randomly select the blue, randomly select the red, but one of each. Then I want to have one randomly selected older female in the group, one randomly selected younger male, and so on. And so now it kind of ensures that we have the same distribution of these confounding factors between the groups.
But you must assume that you know the confounding factors before the study.
Always, because it's a blocking. So every blocking factor must be known. If you don't know it, you cannot block.
Is that realistic?
Well, that's why you randomize, right? So you block on what you know, and you randomize against what you don't know. And usually blocking is effective primarily if these confounding factors have very large effects on your abundances. Because here, instead of having 8 data points, I only have 4 differences. So it has an effect of, reducing degrees of freedom, reducing the number of observations. So if, in fact, there was no day-to-day variation, you would be better off comparing the mean of this group to the mean of this group. And because of that, this type of designs are best done using pilot studies first, where you learn about what are the potential sources of confounding and account for them. Now, our clinical collaborators tell us that age and gender influences everything. So you can pretty much upfront say that you control for age and gender. But then for other things, it very much depends. And usually you don't want to overdo it, right? Because then within each of those blocks, you have a fairly small sample size. So you really want to block for things that are the most important and randomize for the rest.
Sorry, another question. Would you, at this point, consider maybe reducing the complexity of your pool altogether and focus on one subgroup to make it?
That's also very good. That's also very good. So if I go back to my original slide here, what I was saying, right, that there are 2 populations, and we collect data from them, right? The definition of the population is up to you. So if your population is, I want to have all male and female of all ages and all ethnicities, well, clearly it's much bigger burden on you, right, to show that this holds across this big group. So it requires larger sample size, it requires accounting for confounders and so on. Now, if on the other hand you say, I'm only confining my populations to females between this and this age, with this and this lifestyle, and so on, now your populations become much smaller. So typically it's easier to show the differences. But then you have to be very careful not extrapolate this and say that these changes happen to everybody, right? Because your study was narrowed down to a very specific population. But that's very typically done. And by the way, sometimes, especially like in academic research, when we cannot design clinical trials and do things kind of the right way, you just have some samples, you say, okay, I can reasonably assume that they were randomly selected from all the patients who come to this clinic. Now, what is the population that these samples can reasonably represent? And then you kind of work backwards and you say, okay, these samples can be viewed as representative of this population, but they're probably not representative of a broader population. But that's definitely a way to do it. So here's an example from one collaboration that we had, so kind of exactly like that. So we had patients, it was a collaboration with the Munich Heart Center, and they stored plasma samples of everybody who comes to their clinic, and then our collaborators just wanted to have a pilot study for a small number of samples, and that's where they told us age and gender are important, and they stratified for every disease and for age and gender group. These were the number of samples they had in the freezer, and one way to block randomize would be to randomly select samples from the row, making sure that the row has the same counts. So we have the same number of older females across all disease groups, the same number of younger males across all disease groups, and so on. Each row can have different numbers, but we don't want to have control which are all older female and disease which are all younger in male, something like that. So that's a device for that. And then labeling for mass spectrometry is actually also another type of blocking where mass spectrometry run can be thought of as a block. So for example, if we use, let's say we have a SRM experiment where we use labeled reference peptides which are spiked in constant concentration for each endogenous counterpart, you can think again as a run being a restriction on randomization, because within each run you acquire both things simultaneously. And so now if we had a lot of run-to-run variation, let's say we have, these are biological replicates in healthy, this is biological replicates in disease. So if we just compare the mean of the blue to the mean of the red, there's so much variation that we don't see much change. However, these black dots here, they are the spiked labeled references, which are spiked in a constant concentration. So they're supposed to be constant, there's no biology, but we see some changes, meaning that these are technological artifacts. But now if we look at the deviation of blue endogenous from the corresponding labeled reference, we see that these changes are systematically smaller than if we look at the changes of red from the corresponding reference. And so this is another example of blocking. So because these changes are larger than these changes, it points to the difference. And again, remember it's on a log scale, so we're talking about log ratios, right? So that's another example of that. Okay, let me take a minute. Do you have any thoughts on the design or questions?
It's just very clear. We have no.
Oh, okay.
Very clear.
Well, I also talk very fast, so make sure you stop me. Okay. All right. So then, if it's clear, then we will go to the basic data analysis. And again, those of you who are familiar with that, bear with me, because I think it's really important that we're all on the same page about what this is doing. So what I will do, I will talk about a simple t-test and p-values, and I will tell you how good p-values are, and then I will spend some time telling you how bad p-values are. Okay, so that's the plan. Okay, so again, going back to our experimental design, right? So what essentially we're trying to do now is to take what we see, right? So the measurements that we collect, and summarize them somehow and describe their statistical properties so that we can make meaningful statements about the populations. How does it work? So first of all, let me just elaborate a little bit on this fold change versus log fold change and log intensity. Just to clarify, if we think about fold change in an experiment which has replicates, let's say we have 10 healthy and then we have 10 disease, right? And we're interested in the fold change between the groups. The question is, we need to summarize somehow healthy replicates, we need somehow summarize disease replicates and calculate their ratios. So you can think of this as a fold change being some kind of typical value of protein abundance in group 1 and typical value of protein abundance in group 2. Okay, so if I'm interested in log fold change, it's the difference of the logs of these typical values, right? And so now the question is, what is this typical value if we work on the log scale? So here, I promise not much math, this is really an exception, but just to kind of be very precise about what actually happens. So let's say the value y is our log peak intensity. So what I'm doing here, I take the mean log intensity of peaks in group 1 minus mean log intensity of peaks in group 2 for a particular protein, right? We're talking about one protein only, right? So this is the estimate of fold change. So now I'm just rewriting this like fully. I say 1 over number of replicates in group 1, sum of log intensities, minus 1 over number of replicates in group 2, sum of log intensities. So now I spell out the fact that this is the log of the intensities on the original scale minus log of the intensity on the original scale. And so now there's a property of the logs which says that if we have the sum, they can be written in terms of products and powers. And the difference of logs can be rewritten in terms of logs of ratios. So essentially what it means is that if we model our peak intensities on the log scale, we are obtaining log fold changes, where fold changes are geometric means of peak intensities in each group. So if we take the log, if first thing we do, we take the log of peak intensities in the beginning, and then we take the means, compare the means, we get log fold changes. In this log fold change, the typical value in each group is the geometric mean of the peak intensity in each group. And that's really what the relationship is to things, for example, that Andy talked about this morning. Okay, so this is just a note on that. So now let's talk about two-sample t-test, because our question is to know if on average over all the replicates, the mean in the population of healthy is systematically different from the mean in the population in disease. So another way to rewrite this, remember I had those Greek letters. Remember I was saying Greek letters are for things we don't observe, right? We don't see. So another way to rewrite this is to say the mean of the population 1 minus the mean of population 2 is 0. And so those values we never, ever know what they are, right? But it's okay, because what we can do, we can make guesses about them. And that's exactly what we will be doing. So this is our null hypothesis. And the alternative hypothesis is that this difference is actually something else and not 0. So now what we will need to do is to essentially build a signal-to-noise ratio, where our signal is in the data, the difference between the means. Our noise is the estimate of variation associated with these differences in the means. And we will use the signal-to-noise to make some statements about the underlying populations. Okay, so the signal is pretty clear. So this is our log-fold change, where we compare essentially geometric means between each group. So now we need to think a little bit about the estimate of variation. The quantity here is the sample variance of group 1 divided by the number of replicates plus the sample variance of group 2 divided by number of replicates, square root of all of that. Why? Right? So let me just now take it a little bit slower. Actually, let me go all the way here. So this is what's going on. We are trying to characterize the variability associated with the mean, right? So we want to know on the log scale what is the mean of all the replicates in the group healthy and how much uncertainty we have with this mean. Here's an example. And by the way, this comes from a collection of vignettes in nature methods, which are called point of significance. And it's 1 or 2 page summaries on very specific topics and statistics. And this comes from here, and there is a text that describes this figure, so you can go and see a little bit more details. Okay, in this particular, so let's forget about our proteomics for a second. This is a very hypothetical example. Let's say we have a population which has protein abundances which come from this normally looking distribution. We will collect data from this population. Let's say we have sample size of 3 replicates, right? We take 3 replicates, calculate the mean. Now we take 3 more replicates, calculate the mean. You want to repeat the experiment again, 3 more replicates, calculate the mean. And do it a bunch of times. And now let's draw the histogram of the means. So this is what the histogram of the means would look like. So if we take 3 replicates, calculate the mean, repeat the experiment, 3 replicates the mean, start over, 3 replicates, repeat the mean. We see how much variation is there between the means. We draw this histogram, and this is what it looks like. Now, if we do, instead of 3 replicates, we do 5 replicates. This is what the histogram would look like. If we do 10, that's what it looks like. If we do 20, that's what it looks like. So essentially, you can see that as the sample size increases, our means become more and more reproducible. So this histogram becomes more and more narrow, right? And every time we repeat the experiment, we get the value which is more and more similar to each other. This histogram is called the sampling distribution of the mean. That's how much uncertainty we have in the mean. The variability in the underlying distribution is characterized with the sample variance or standard deviation, right? And the variability of this histogram is the sampling distribution or sampling variance of the mean. So this is a really important distinction, right? So standard deviation or variance describes how much variability is in the population. Sampling variance of the mean describes how much uncertainty we have in the mean of some number of replicates. And so now, if I go back to my formula, so this was the variance in the population. The variance divided by the sample size is the variance of these distributions. And so now we see that if we want to construct the noise associated with the signal, we'll say, okay, how much variability we have associated with this mean? how much variability we have associated with this mean, and we take square root to bring it on the same scale. So the square root of this quantity is called the standard error. So again, just to make sure we distinguish, this is the standard deviation, and the variability here is the standard error. And therefore, this signal to noise tells us this is how much signal we have in the means, this is how much uncertainty we have in the means. Interestingly now, if we start from something else, and not just bell-shaped distribution, but any other distribution, we can see that as the sample size increases, our histograms become more and more bell-shaped. If we start from something really weird, so the width of this histogram is not as narrow as here, so here is narrower, but it still looks quite bell-shaped. This is called central limit theorem. So it says that as we have more number of replicates, the uncertainty in the mean becomes roughly normally distributed, even if the population itself is not distributed normally. So here, people often ask me, okay, what if my data are not normally distributed, right? What if even on the log scale I have some kind of outliers and so on? And the answer is, it's okay, because we're interested in the means and not in the individual numbers. And if we have enough replicates, the means will be approximately normally distributed, even if the individual samples are not quite normally distributed. Does it make sense? So now we can construct this signal-to-noise ratio based on the understanding of what it means to have noise in the means, right? So this is the sampling distribution. Okay, so I'll skip that. And so now what we need to do is to essentially, yes, please.
Usually when we have normal distribution, we think mean is better to take, and then when we have normally distributed, the median is better. So in this example, these examples, so you're saying that when you have more number, your mean and median are becoming the same?
That's right, because so means equal to the medians when the distributions are symmetric, right? Even if they're not normal, as long as they're symmetric, it will be the same, right? And the central limit theorem says that, actually, well, the full thing is, if it starts from the normal, this is exactly normal. If it starts from any other distribution, this is approximately normal. Therefore, even if you start from something very skewed, and if you have enough replicates, the means themselves will be roughly symmetric, and the means will be similar to the median of this distribution. The problem is that if you start with something very skewed, it takes more replicates to get to this point. This is why we take the log, because on the log scale it's more symmetric, and so you get closer to the right properties faster. And it helps in terms of sample size. Okay. Right, so now we constructed the signal to noise. We can actually calculate this from the data. And the last question is, okay, clearly we want signal to be larger than noise, right? But how large is large? So what is our cutoff to say, okay, this signal is much larger than the noise? And this is where this assumption of normal distribution comes in place. Because if things are exactly normal or approximately normal, as in the central limit theorem, we can mathematically, without any, additional things, we have to assume that each replicate is independent. But other than that, we can mathematically describe the plausible range of values of the signal-to-noise ratio if, in fact, there is no difference between the populations. Okay? So if there is no difference between the populations, right? We don't know what they are, but we can assume, right? Assuming that there is no difference between the populations, and building signal-to-noise in this way, we can mathematically describe what the range of the signal-to-noise would be. And it's described in this distribution, which is bell-shaped. So it is approximate to normal distribution. And probability distribution, the property is the area under the curve is the probability of things happening, right? So now what we can do, we can essentially have the cutoffs such that the probability to the left plus probability to the right is as small as we would like. And this is called false positive rate. This is alpha. This is our threshold. So let's say alpha is 5%. So we will have 0.025 here and 0.025 here. This is a small probability that we see something quite extreme, when in fact there is no difference between the groups. And so now what we will do next, we will actually calculate the signal-to-noise ratio from our data and overlay this on top of this graph. So if our calculated signal to noise is in the center, we'll say, yeah, well, that's what's expected in the range of values if there is no difference between the groups. Therefore, there is no evidence of difference between the groups. On the other hand, if the signal to noise is large, let's say group 1 has the protein highly upregulated, we will have some value which is quite far away. This is 0 in the center, so quite far away from 0. Then we say, wait a minute, this is a very unusual value for this expected range of values, right? So what happens? Well, our assumption that there is no difference between the groups is wrong. Therefore, it's evidence against this null hypothesis. It's the evidence in favor of the alternative. And we reject the null hypothesis. So essentially, the assumption of independence and normality, or approximate normality, allows us to establish those goalposts, which say, well, outside of this range of values, it's too large to be, consistent with the null hypothesis. That's really the value of that. And so the very last kind of related concept is the p-value. So one thing is we can just say, okay, these are our cutoffs, but on the other hand, we can actually overlay this signal to noise on this graph as is, right? And because we don't have preference in any direction, we overlay that, and we overlay the symmetric counterpart around that. And so now the area to the left of this vertical line plus the area to the right of this vertical line is the p-value. And so if the p-value is smaller than alpha, it means that we are in the rejection region, right? We're outside of these goalposts. If the p-value is larger than alpha, it means we're inside the goalposts, and we don't have evidence against null hypothesis. That's it. Okay? So really what we're doing is having the right way to calculate signal-to-noise ratio, and then make assumptions about the data. And the assumptions are that the means are approximately normal and the individual replicates are independent, to establish a goalpost, how large the signal-to-noise has to be to claim that there is evidence against the null hypothesis. Okay? All right. So the problem with p-values, I'm still in the kind of part where I talk about how good they are, right? But like a slightly kind of disadvantage of p-values is that once we have a p-value of 0.00 something, we don't really know what's the extent of change, right? Is it up and down, and how much uncertainty was in this change, right? And so it is useful to list a whole bunch of proteins and annotate them with the p-values. But if we want to say something meaningful about the underlying biology, we lost a lot of information. A really good alternative to represent the results of this type of things is confidence interval, or error bars, or interval estimation. And we already talked about two things. So for example, we can say it's the signal plus minus standard error, right? So remember again, the standard error is the square, it's essentially the denominator, right? It's our noise. Or if we have one sample, standard error is the square root of the variance of the sampling distribution. So we can do that. We can also do the signal plus minus standard deviation. So standard deviation talks about the actual population, how much variability is in the actual population. The third type of error bar, which we haven't talked about yet, is the confidence interval. And so in fact, when we talk about error bars and interval estimation, we have at least three choices, right? Plus minus standard deviation, plus minus standard error, and confidence interval. So standard deviation describes the population. Standard error describes how much uncertainty we have in the mean. Confidence interval adds a probabilistic interpretation, so it adds actual level of confidence in that. Confidence interval is actually not an interval, it's a procedure. And the procedure is such that if you follow this procedure, let's say 95% confidence interval, if you follow this procedure and you collect the data 100 times and apply this procedure 100 times, 95 intervals will contain the true value. And 5 intervals will be outside of the true value. And again, the true value in the population, right? So here's an example. If we have just one, let's say healthy, right, some kind of distribution, this is the true mean in the population. We keep repeating this procedure and building it over and over. We would expect 5 confidence intervals not covering the true value and 95 covering the true value. And this probabilistic interpretation comes again from exactly the same argument. We can describe the property of signal to noise, and then we just invert the signal to noise. So this is our signal, plus minus our noise, and this multiplier is the value of this goalpost. And because this goalpost have, let's say, 5% in the tails, it means that this procedure will also have 5% of chance of having confidence intervals not covering the true values. And again, just like before, if we assume independence and approximate normality, we can mathematically describe the distribution. Here we can mathematically derive this procedure. So essentially it comes from a mathematical argument. And so now here's an example comparing standard error versus confidence interval. The gray lines here is the mean plus minus standard error. The black line is the mean plus minus confidence interval. And you see that confidence interval is always quite a bit wider than standard errors. And of course, both become narrower and narrower as sample size increases, right? Because sample size is in denominator. So it becomes smaller and smaller. Just to give you another overview of that, again, from the same collection of vignettes, here's an example. So they have sample size of 10. They adjusted the differences between the means, the difference between the means is fixed, they adjusted the variabilities so that the three types of error bars look exactly identical. And you see that this corresponds to very different p-values. On the other hand, they adjusted this to have identical p-values, and you see that the appearance of the error bars is super different depending on which kind of error bar it is. And then they show how it depends on p-values for the standard error and how it depends on p-values for the confidence interval. So essentially, the points are that different intervals indicate different things. They describe different aspects of the data. And because of that, the associated error bars can look very differently. And for example, this may look like not a big difference between the groups, when in fact the conclusion is identical to these error bars. But these error bars describe a different property of the data. So at the very least, when you report error bars, document what they are, because the interpretation really depends on what it is, right? And on the other hand, don't be afraid of error bars that overlap if these are actually confidence intervals, because it may still mean a relatively substantial change. It's okay? I'll let you breathe for a second. Okay, so another really important concept in statistics is statistical power. And this is something that we care about a lot when we design the experiment. So this is really the concept we optimize for. Statistical power is the probability of finding a difference when the difference exists, right? Because we do things hoping to find some true changes, right? And so this is actually mathematically probability of finding the change between conditions when this change is actually present. So if I bring back this is called reference distribution. So let's say we know that likely the change will be upregulated. We can put all the alpha to the right tail. So we have this distribution of signal to noise. So if we are in this region, there is no evidence against the null hypothesis. If it's in this region, we reject the null hypothesis. And this is what happens if, in fact, there is no difference between the groups. However, if between the groups the proton is, in fact, regulated, and if, in fact, there is a difference between the groups, this plausible range of values of the signal to noise will look differently, and it will be shifted away from 0, right? So let's say group 1 has systematically larger protein abundances than group 2, so the signal to noise will tend to be larger than 0. But this is where we make this cutoff, right? Because this is our decision is based on the null hypothesis. So the statistical power maps this cutoff on the distribution if, in fact, there is a difference. And it's the area to the right of this cutoff for the distribution when, in fact, there is a change. So to calculate the statistical power, we will need to know what is the plausible range of values of the signal to noise. And we will need to know a lot of things. We will need to know alpha, for example, right? But most importantly, we would need to predict what is the actual regulation between the groups. We would need to predict how much variability we have in each of the groups. So calculating statistical power is, again, something which could only be done if you have a pilot experiment which tells you already how much variability we can have in our system. And of course, the larger the regulation, the easier it will be to reject the null hypothesis, because the bigger the signal would be. So one really bad choice to gain power is to have larger alpha and say, I will reject null hypothesis for everything where the p-value is below 0.1 or 0.15 or 0.2, because then we have a lot of false positives. A better way to increase statistical power is to shift this distribution of plausible values of signal to noise to the right. And we can do it, for example, when the protein is more regulated, but we can also do this by increasing the sample size.
Would you recommend every time to give this mean should be the geometrical mean?
Well, so that's yes. The short answer is yes. And in fact, at the end of the day today, you will be working with MS Stats. This is the tool that we developed for that. And the very first thing we do, we take the log of peak intensities. And if you take the log of peak intensities, everything else follows, including when at the end you arrive to log fold changes. You know that your fold change is represented by a geometric, kind of, glorified version of geometric means in each group. And glorified because we take care of outliers, we take care of missing values, and so on. But essentially, these are geometric means in each group. Okay, so just to summarize here, so a better way to increase statistical power is to shift this distribution of plausible values of signal to noise if, in fact, there's a difference between the groups. And we can do it, well, by focusing on really large changes, right? If we work with systems which have a lot of regulation, it will be easy to detect them. That's kind of easy. But another is to increase sample size or decrease the variability in the means, right? And so this is what gives us statistical power. Okay, and this is just a summary of sample size calculation. So, the most frequent question that people ask me is, how many replicates do I need, right? Can I use, my grant is due tomorrow, can I use 3 patients? Right, something like that. There's a really funny video, statistician talking to a life scientist, and they could not understand each other at all. So it comes from that. So, and essentially, if you come to me and say, can I use 3 patients? I will say, well, who knows, maybe, it depends, right? And it depends on what? So it depends on many things. It depends on alpha, right? So this is where my goalpost will be. The more towards the tail is my goalpost, the more difficult for me it will be to have high power. It is also probability of false negative. So false negative is here. It is saying, well, if in fact there is a change, how likely we are to actually not discover this change. It is the anticipated log fold change. And usually what people do, you would just say, this is the smallest fold change I'm interested in. If it's below that, it's just practically not very useful to me. And you just specify the smallest value. So these 3, we can specify them from common practice, from literature, and so on. The biggest challenge is the biological and technological variation. So this is this quantities which go into the variability in the actual populations. And those need to come from as similar workflow and as similar biological material as possible to the study that you plan. And the best way to do this is to have pilot studies where you get some sense of how much variability is there. And then, well, kind of the technical way of doing is there is essentially a formula which is very easy to derive mathematically, saying that my noise, right, so the variance in the denominator has to be bound by a quantity which is derived from that, and you can solve for sample size, you can solve for the smallest fold change which can be detected given the sample size and so on. And you make figures like that, and we do this in MS Stats, you will see. So the x-axis is the fold change, and the y-axis is number of replicates per group. And well, clearly the smaller the fold change is, the more replicates you need, right? But you can also think in terms of changing power or changing alpha and so on. So that's how the calculation will work. So this is how much we need to know to actually be able to design the experiment for the future study. Okay, let me take another pause. Any questions so far?
ratio and then?
Once you take the log, so the whole point of log is to not do ratios, right? So once you take the log, you look at differences. And what you will do, kind of going back to my figure here, so these are log intensities, right? So you will have the log intensities of standards, log intensity of the endogenous. And, well, one simple way to do this, look at differences of endogenous versus standards for healthy, endogenous versus standards for disease, and then compare the differences in this group to differences in this group. So now your input to this two-group situation will be deviations from the references, right, as opposed to the actual intensity. And you proceed exactly the same way. And we do this in MS Stats, but we do it in a more complicated way because also we take care of missing values, outliers, complex designs, and so on. But that's the idea. Okay, so then the next thing is, so I promised I will tell you things to be careful with, especially when we talk about p-values. So p-values have received a lot of somewhat negative attention recently, to the point that some journals say we will not publish p-values anymore. It's true. And there are some good reasons for that. And I'm not saying not to use p-values, but I'm saying it's really important to be aware of the limitations of the p-values. For example, people sometimes say, I want to design study to make sure I have a very small p-value. Or they will adjust the analysis protocols to make sure that they get a very small p-value. And these are really not good ways of doing it. So here's the reason for that. So this is another and a perspective paper from Nature Methods, which illustrates the fact that because p-values are derived from data, and because data are random, right, they have randomness and variation, therefore p-values themselves are random variables. Therefore, p-values, we cannot expect the p-values to always be the same if we repeat the study over and over. And here they just have a simulation example, completely fake data, two populations, small difference between the two groups compared to the variability in each population. What they did, they took 10 replicates from group A, 2 replicates from group B, did the t-test exactly as we just described. They found pretty large difference, p-value very small, fold change 1.46. And then I did this again, exactly the same procedure. So this time they found p-value 0.82. And they changed in the opposite direction. They did this again, found p-value 0.85. The change is again going up, right? And here the p-value is borderline. So you do this 4 times, and especially in this situation, with a sample size of 10, from one iteration of experiment to another, you can get completely different conclusions. So on one hand, if we have a p-value that is not in the significance range, it may be that we do not have enough power to reject a null hypothesis. On the other hand, if we have statistical significance, it may be an artifact. So p-value is not the last judgment call about what we do. And to illustrate that, they did more simulations like that. Instead of doing it 4 times, they actually did it 1,000 times. And now here, they make a histogram of p-values. They take a log of p-value just so that we can focus on the region where the p-values are small, right? That's where we're usually interested in. And again, for these 2 populations, they made a histogram of the log p-values. And you see that out of 1,000, only 186 are in the rejection area. The vast majority of p-values are in the large area, even though there is a true difference between the groups. Why? Because probably here, with a sample size of 10, this is a study that is underpowered, right? So this is the study where with sample size of 10, this distribution under the true difference between the groups didn't shift to the right enough. So this blue area is too small. So we have a very small probability of detecting the change when the change exists. So now they do sample size 30. You see that the histogram shifts a little bit. So now you have 469 experiments which are in the significance area. Sample size 64, we have 800 experiments in the significance. And it's only with sample size of 100 when you get the vast majority of iterations consistent and same results. So if we have a small sample size and a small p-value, really doesn't mean much. So it can really go in both directions, and oftentimes we don't have enough power, and sometimes it can be just an artifact. And this is to illustrate that. So this is further, this is again the same thing, 10, 30, 64, and 100 replicates. The x-axis is the same as before with the log of the p-values. The y-axis is the reported difference between the groups. And so now, kind of depending on the sample size, you see here p-values tend to be large, and then sample size increases, p-values tend to be smaller. The most interesting thing here is this histograms. So the y-axis is the reported difference between the groups. Here, they plot the difference between the groups, but only among significant experiments. So essentially, what they do is they get the data, do a t-test. If it's significant, report the difference. If it's not significant, don't report it, right? Do it again, right? Make a hypothesis testing. If the difference is significant, report the p-value. Otherwise, do not report. And so now these histograms, they're histograms of things which they chose to report based on significance. So now the true difference is 0.05. The y-axis is the reported difference everywhere. This histogram are the reported difference among experiments which survived the p-value cutoff. And so what you see, and to me this is really striking, that not a single reported difference is even close to the truth. It's always vastly exaggerated. So when the sample size is small, when the experiment is underpowered, you are likely to not find the difference. But worse, if you do find a difference, you are very likely to overstate the extent of regulation which is associated with the conditions. And it's only when we increase the number of replicates that things become roughly stable and roughly varying around the true value of 0.5. So this is not to say that p-values are bad. This is to say that they are random variables, and we need to understand their properties. And therefore, we should not be making decisions for follow-up, or for publishing, or for, making big claims if we have small-scale studies and based on the p-value as the ultimate evidence. That's really important. Okay, so I have some other things, but let me first answer the question that you asked. So this morning about pooling, right? So I don't have much on pooling here, but I think now it may be a good time to mention that. So the question was, if we have 10 replicates in group 1 and we have 10 replicates in group 2, can I pool all 10 replicates in group 1 together? Can I pool all 10 replicates in group 2 together? And then maybe have 3 technical mass spectrometry runs from the first group and 3 technical mass spectrometry runs in the second group. What do you think of that? Why?
I mean, because of everything that you've been saying, right? Because if you pool, you completely lose your biological replicates. So
Thank you.
Have 3 biological replicates instead of 30.
Exactly. So you have your pool, and you measured it several times, so now you quantify this pool really, really well, but they're all together. You don't know how much person-to-person variation is there, right? So this is the thing not to do, right? If you need to pool, now pooling is not a bad thing, right? So pooling is good, but that particular pooling is not good. So what you can do if you have 10 biological replicates, right? If you have a pool of, 2, another pool of 2, right? So now you reduce the number of runs to 5, where each of them is a pool of 2. You do the same thing for the disease. You can still get person-to-person variation based on these pools, provided that each pool is independent, right? You don't have the same sample in multiple pools, right? And that is a good strategy. And in fact, theoretically, it can be shown that pooling is better than non-pooling, given the fixed number of runs. But there are also practical limitations. For example, if one sample in the pool is compromised, the entire pool is compromised, right? So if your goal is predicting the disease status for biomarker discovery, now you have, but they're all together. You don't know how much person-to-person variation is there, right? So this is the thing not to do, right? If you need to pool, now pooling is not a bad thing, right? So pooling is good, but that particular pooling is not good. So what you can do if you have 10 biological replicates, right? If you have a pool of, 2, another pool of 2, right? So now you reduce the number of runs to 5, where each of them is a pool of 2. You do the same thing for the disease. you can still get person-to-person variation based on these pools, provided that each pool is independent, right? You don't have the same sample in multiple pools, right? And that is a good strategy. And in fact, theoretically, it can be shown that pooling is better than non-pooling, given the fixed number of runs. But there are also practical limitations. For example, if one sample in the pool is compromised, the entire pool is compromised, right? So if your goal is predicting the disease status for biomarker discovery, now you have, but they're all together. You don't know how much person-to-person variation is there, right? So this is the thing not to do, right? If you need to pool, now pooling is not a bad thing. Absolutely, I'm just trying to get rid of this thing. I'm not sure how to do it. No, hopefully it will go away by itself. It's very, okay, let me just, hopefully somebody will come. So, okay, good. Here, for example, right? So this is our variability, right? So essentially you have to bound this entire term by some constant, right? So now this term can be reduced in many ways, right? For example, if you assume the same number of replicates in each group, you just pick one that, right? However, if one group has much larger variance than the other, you can say, okay, I will probably have more replicates from the larger variance group, right? And fewer replicates from the smaller variance group. Or if it turns out that the larger variance group is very expensive samples, which are very difficult to find, this will give you an idea of how many more controls you would need to be able to reduce this entire term. So there are really ways to play with that to some extent. But again, this would require you knowing what these variances are in the first place from some kind of pilot studies. Okay, so this is the p-value. So now one more thing about p-values, which I have not mentioned yet, is the fact that they refer to one protein. And of course, we in our studies don't have one protein. We have many more proteins, right? And so now we are also in a situation where we have many, many, many p-values for many proteins. So how do we deal with that? And it's an issue. So for example, here is an example from the blog also from Jeff, who was talking about somebody's poster who was studying with MRI the brain activity of dead fish. And they found many active regions in the brain of a dead fish, right? Brain fish of dead fish has activity, right? Sounds strange, but again, remember the way this process works, we set aside 5% of probability that we reject the null hypothesis when in fact it is true, right? So this is some non-negligible probability. So if you have 100 proteins, you will have on average 5 false positives. If you have 1,000 proteins, you will have on average 50 proteins which are false positive, right? So this is just how this is set up. So we do expect certain amount of false positives if we have many, many tests over many proteins. So how do we deal with that? Well, this is an illustration. Let's say we just have 2 proteins, just to be specific. So we have this now distributions of signal to noise, which we use to define rejection region. Let's say protein 1. We have this distribution. We set aside alpha, and we reject the null hypothesis in this or in this region, regardless of what happens for protein 2. So now this area plus this area is 5%. We do the same for protein 2. So this area plus this area is also 5%. But the fact is that we have both proteins in our study, so the probability of making at least one mistake, right, so at least one false positive, it's actually a combination of both. And the combined area is, of course, more than 0.05. And so now if we grow the number of proteins, the probability of making at least one false positive is essentially 1. We are guaranteed to have at least one false positive in our study. How people deal with that? Well, the easiest way is essentially to take this decision boundary and push it back, kind of like that, so that the area outside of the dashed line is 0.05 or whatever we want to do.
Could you repeat this last?
So is this clear? So for one protein, we have our rejection region. The probability of false positive is alpha, right? Because we say this is the area under the curve is the probability to claim we reject the null hypothesis when in fact null hypothesis is true. So these are false positives, right? And we do it no matter what happens for protein 2. We do it for protein 1. Same thing for protein 2, right? So we compare this to this reference value, reject the null hypothesis with probability 5% of false positive, regardless of protein 1. But both proteins are in our study. So probability of at least one false positive in at least one protein is the union of these two areas. And so the probability of this combined area is more than 0.05, right? And so now we have bigger, we keep accumulating this false positive rate, right? And so if we have a DIA experiment with 10,000 proteins, the probability of at least one false positive is, you're guaranteed, right? So how do we deal with that? And essentially we want to control this combined red area here to be 5% or whatever we want it to be. So one way to do this is to essentially make our cutoffs more conservative, so that the area outside of the dashed line is 5%. And this is exactly what is done, and this is state of the art, except that how exactly we define this dashed line, it depends on the method, right? And you probably have heard about Bonferroni approach, right? Which takes alpha and divides by the number of proteins. And this will control the area outside of the dashed line, except that this becomes super conservative. If we have 10,000 proteins, right, and we take 5%, divide by 10,000, your rejection region varies very, very far in the tail. And I like to say that the best way to not make any false positives is to not find anything. Then we're sure that we have no false positives, but that's not very productive, right? So we need to do something else. So Bonferroni is not a really good approach for our experiments, but you probably have heard of that. That's exactly what it's doing. A better way of doing that is to control false discovery rate. And now this is false discovery rate of hypothesis testing. It's orthogonal to FDR, in identification of MSMS spectra, library research, and so on, right? It's completely orthogonal to that. And so false discovery rate works with a table like that, where this is the truth. How many proteins are truly not changing? How many proteins are truly changing? And this is our decision. How many we claim as not changing? How many we claim as changing? And the fact is that the only thing which is observable in this table is R, or M minus R, right? So this is how many changes we claim and how many we don't claim. But just like before, we can make a statistical argument to essentially, with some assumptions, we can characterize the average behavior of this table. And so keeping our probability of at least one false positive very small is the same as keeping the probability of V being anything but 0 very small. So that's what Bonferroni is doing. But false discovery rate will say, I'm fine with V being 1, 2, 3, 4, 5, whatever, as long as I can control the proportion of V in R, right? So the proportion of false positives in the full list of rejections. And so false discovery rate is actually the expected ratio of V over R. So here, just to avoid division by 0, we take that, but we always find some differences, so you can just think of this as V over R. Expected means that if you keep collecting the data and repeating this procedure, on average, V over R will be as we would like it to be. But again, it's a random quantity, so it may have variability between experiments. And so a typical way to report that, and this is what we do in MSStats, is to report for many proteins the x-axis, which has log of the fold change. That's what we call the practical significance. This is the p-value adjusted such that the cutoff of the adjusted p-value controls false discovery rate. And we take the log just so that high values correspond to higher statistical significance. And so now the horizontal line corresponds to the FDR cutoff of whatever you would like. And so what's interesting to us is the top left and top right corner. So they're both statistically significant and practically significant. And it's totally fine to apply a fold change cutoff, provided that it also comes with a statistical significance cutoff. Okay. Oh, and so that's important. So now we can also adjust our sample size calculation to the case where we have more than one protein. If we have one protein, we discussed already what happens, right? Smaller fold change means larger number of replicates. MSStats produces curves like that for experiments which have many proteins. Now, the solid line here is as before, if we only had one protein in the experiment. But this dashed line corresponds to experiments with many proteins. The middle line, if we expect that half of the proteins are changing, for example, in a targeted SRM experiment, the dashed line corresponds to discovery experiments where we expect very small, like 1% of proteins changing. And so what we see is that the experiments where only few proteins change require larger sample size. And this is something which is often overlooked because essentially the idea is that we want to protect ourselves against false positives. If everything is a true positive, we don't need much protection, right? But if there are many opportunities for false positives, we need to work harder to control the overall false positive rate at some level. And work harder means have a larger sample size. Okay, so how about I'll take a break here. I think it's more or less the time now, right?